Good morning, everyone. So, today we are going to start with the new topic, that is bioavailability and bioequivalence study. You have already studied about these concepts in your general pharmacology topic, that is one that was the part of the pharmacology one. Okay. So, let's see, it is actually only introductory part over here. So, as it is part of preclinical and clinical development, that's why we have to study what is bioavailability and bioequivalence studies. Okay, so introduction. So it is essential to ensure in uniformity in standards of quality, efficacy, and safety of pharmaceutical products. Okay. So, as per this sentence, why we conduct bioavailability and bioequivalence studies in order to ensure uniformity. Okay, so uniformity in what? The standards of quality, efficacy, and safety. Okay, so whenever these bioavailability and bioequivalence studies are carried out, they ensure that whatever safety, quality, and efficacy studies we carry out of the pharmaceutical product, they are of certain quality and they are uniform in nature. Okay, so there should not be any deviation, any variation among the various studies which are carried out among on the pharmaceutical products. Okay, second thing, that they give reasonable assurance that is to be provided that various products containing same active ingredients marketed by different licensees are clinically equivalent and interchangeable. Okay. So these studies, they provide us the assurance that even though the same API containing products or two different brands by the two different companies, they have the similar clinical effect, not the same, but the similar clinical effect, and it can also be used interchangeably. Okay, so example of this is that whenever uh, we go to the medical shop, and if whatever written into the prescription, if it is not available with the medical shop, then by consulting with the physician, the pharmacist can give other uh, drug or other brand of drug. Okay, so when he can give other brand of drug, when he knows that this other brand of drug is equivalent, bioequivalent to that of the first brand of drug. Okay, so these studies, bioavailability and bioequivalence studies, they ensure that they assure us that even if we take another brand of drug, then it will produce the clinical effect to the same extent. Okay, so next is the release of an active substance should be known and reproducible. Okay, so the disintegration and the dissolution study should be carried out, and they this disintegration and dissolution rates, they should be similar among the different brands of product. Okay, both bioavailability and bioequivalence focus on a release of drug substance from its dosage form and subsequent absorption in the circulation. Okay, so what is the focus of both these studies is that release of drug from its dosage form. Okay, and how it enters into the systemic circulation. Okay. The next is similar approaches to measure bioavailability should be followed in demonstrating bioequivalence. Okay. So next, let us define what is bioavailability. So it is measurement of the relative amount and rate at which the drug from administered dosage form reaches the systemic circulation and become available at the site of action. Okay, so two things are important over here. It is amount of drug that is getting released from the administered dosage form, and the second thing that is important, that is rate at which this drug is getting released. Okay, so we can consider two examples why these two terminologies, that is amount and rate, are important. First thing is that why we require amount is that if the rate of drug release is faster but the amount of drug that gets into the systemic circulation is less, then also it means that the clinical effect which we get, it will be less. Okay, we will consider the other part of this is that rate at which the drug is getting released. If drug gets released at very faster rate but the amount which gets into the systemic circulation, if it is less, then it means that the clinical effect in this condition will also be less. Okay, so that's why we have to consider both, that is amount which enters the systemic circulation and which reaches to the site of action, and the rate at which the this amount is being released into the systemic circulation. Okay, so there will be two things over here, that if the drug is being given by the oral administration, then these two things they matter the most. Okay, in case of the intravenous or parenteral administration, bioavailability is 100%. Why? Because we directly put the drug into the systemic circulation. There is no question of drug getting disintegrated or dissolution is happening. Okay, as we are putting the drug directly into the systemic circulation, that's why the bioavailability is 100%. Okay, so bioavailable fraction, which is denoted by the letter capital A, it refers to the fraction of administered dose that enters the systemic circulation, and it is calculated as F is equal to bioavailable dose and the administered dose. Okay, although the formula is really simple, but to determine bioavailable dose, we have to do lot of things. Administered dose, it is a very simple thing, that how much drug you give to the patient, that will be the administered dose. So let's take very simple example of the paracetamol. We know the dose is 500 milligrams. So in case of paracetamol, the administered dose will become 500 milligram. But we have to determine what is bioavailable dose. So we have to collect the blood at certain time points, that is at 0 minute, at 30 minutes, at 1 hour, 2 hour, 4 hour, 8 hours, 16 hours, 32 hours, like that. Okay, and that time we have to determine the amount of drug in the in that particular blood sample, and we once we determine it, the amount of drug into that blood sample, we have to uh, multiply it with the amount of the blood. Okay, so it will give us the bioavailable dose. Okay, so it takes much effort to determine bioavailable dose, and thus we can get the bioavailable fraction. Okay, so therapeutic relevance of this concept is that you can see the diagram over here. It is on the x-axis we have the time, and on the y-axis we have plasma drug concentration. Okay, so this type of graph is applicable to the orally administered drug. So apply as we apply the as we give the drug at 0 minute, the concentration is also 0. Okay, so during the absorption phase, the concentration of drug into the plasma, it goes on increasing. At certain level, it will reach the minimum effective concentration, and that's where we will see the onset of action of the drug. Okay, so it will go on increasing, and at some point it will also cross the maximum safe concentration in the plasma. Okay, so at the peak, it will, the concentration which corresponds to the time, it is called as Cmax. Okay, so the, at this particular point, peak plasma concentration, the absorption rate is equal to the elimination rate. It means that the rate at which the absorption of drug is happening is the same at the rate at which the elimination of drug is happening, and the time at which the peak plasma concentration is achieved, that is Cmax, it is called as Tmax. Okay, so post Cmax, the concentration of drug in the plasma is on decline. Okay, so we consider that is post absorption phase. In this in this particular phase, the rate at which the drug getting absorbed is less than drug at which the drug is getting metabolized and distributed. Okay, so distribution rate and the metabolism rate is maximum in post absorption phase as compared to the absorption phase. Okay, so as the drug get distributed and it gets metabolized, the concentration of the drug gets down below the maximum safe concentration, and at certain point it also goes below the minimum effective concentration and will reach into the subtherapeutic level. And when the concentration of drug reaches to the subtherapeutic level, we can call it as the elimination phase. The drug is starting to get eliminated from the body. Okay, so at that, at this particular time, if we administer the next dose of the drug, then again the concentration of the drug in the blood plasma will also be on the rise instead of it is declining. Okay, so there are two concepts of bioavailability. That first one is absolute and second one is relative. So what is absolute bioavailability? It compares the bioavailability of the active drug in systemic circulation following non-intravenous administration with the same drug following intravenous administration. So we are comparing the bioavailability of the drug which is given by the oral route or any other route apart from the intravenous route, and divided by the drug which is given by the intravenous route. Okay, so here we calculate the area under the curve for the drug which is given by the non-intravenous route, and we divide that area under the curve by the area under the curve of the drug which is given by the IV route. And when you can see in the formula, when this AUC of the drug given by the non-intravenous route is divided by the AUC of the drug given by the intravenous route, we get the absolute bioavailability, which is denoted by a letter that is F to the subscription, that is AB. AB stands for absolute bioavailability. Okay. So what is the relative bioavailability? It compares the bioavailability of a formulation A of a certain drug when compared with another formulation B of the same drug, usually an established standard. Okay, so this is specifically applied in case of the generic products. Okay, so you know what are generic product? They don't have the brand name. They are sold by the name of the active pharmaceutical ingredient which is incorporated into it. Okay, so we have a standard product. Let's assume a branded product. Let's assume crocin. Okay, so we will determine the bioavailability of crocin and will compare it with the bioavailability of a paracetamol, which is a generic product. And whenever we compare this, and then we will get relative bioavailability. Okay, so the value should come out to be closer to the 1. If it exceeds than the 1, that it means that the bioavailability of the generic product is better than that of the branded product. Okay, if it is 1, that it means that both the products are equivalent in nature, and if it is less than 1, it means that bioavailability of the generic product is less than that of the branded product. Okay, so next, factors that affect the bioavailability of the drug. So there are many factors that we are going to study in um, brief, as you have already studied these factors in many of the other subjects. Okay, so first is physical properties of a drug. So in that first one, that is physical state, liquids, they are more bioavailable as compared to the solids. So in liquids also, the uh, bioavailability fraction is in the descending order as it is shown. That is, it is highest in solution and it is least in coated tablets. Okay, then in crystalloids it is more as compared to in colloids. Okay, so lipid or water solubility. So aqueous phase is at the absorption site. So depending on what is the nature of the drug, its absorption also depends. Okay, so at the absorption site, the it is water phase. So uh, if the drug is highly lipophilic in nature, then it becomes very difficult for such a drug to make contact with the absorption site. Okay, so we have to make certain changes into it so that its absorption will increase and also its bioavailability will increase. Okay, the next is second factor, that is dosage forms. Okay, so in that first one, that is particle size. So it is important for the sparingly soluble drugs. So if we decrease the size of the particles, then the absorption increases and we have to decrease the dose of the drug in this condition. Okay, so example is given of the nanocrystalline formulations of the saquinab. Okay, so by making the nanocrystalline formulations of saquinavir, we are able to decrease the dose of this antiviral agent. Okay, so if specific requirement says that we have to decrease the absorption of the drug, in which condition we have to decrease the absorption of the drug? If we want the drug to remain in the gastrointestinal tract for treating the some local conditions, in that case we have to increase the, sorry, we have to increase the size of the particles. Okay, this, the drug will remain in the gastrointestinal tract and we will get the localized effect. Okay, so next, physiological factors. In that first one, that is ionization. Okay, so we have already studied about ionization. The unionized form penetrates GI mucosal lining very quickly. Okay, because they don't have any charges on it, and as they don't have any charges on it, they don't get repelled, okay, from the surface. Okay, so next is pH of the fluid. In case of weakly acidic drugs like aspirin and barbiturates, stomach and duodenum is the ideal site for the absorption. In case of weakly basic drugs like pethidine and ephedrine, small intestine is the ideal site. And in case of strongly acidic or basic drugs, which are targets for the ionization, they get poorly absorbed if given by the oral route. So in such cases, we have to give the drug by the parenteral route. Okay. Next factor that affects the bioavailability is gastrointestinal transit time. Okay, so there are fewer conditions which are to be which affect the bioavailability. That is, first one is prolonged gastric emptying time. So it delays absorption due to stasis. Okay, so there are certain drugs like anticholinergics, they are known to cause this type of effect. And also in certain complications of a certain disease like a diabetic neuropathy also has this effect on the gastric emptying time. Okay, so in both of these conditions there is decrease in or there is prolongation of gastric emptying time, which is because of decrease in the peristaltic activity of the stomach. And also this increases the gastric emptying time, and this increases the absorption of drugs which get absorbed in stomach, and decreases absorption of drug which are supposed to get absorbed in the small intestine. Okay, so next is increased peristaltic activity. Example of the drug that increases peristaltic activity is metoclopramide, and it is known to speed up the absorption of analgesic when given simultaneously. Okay, the next effect is excessive peristaltic activity, which is observed in diarrhea. It impairs absorption as during diarrhea, it doesn't allow the drug to remain in contact with the gastric mucosal membrane, and as drug cannot stay in the gastrointestinal tract for a prolonged duration of time, its absorption is impaired. And as its absorption is impaired, the bioavailability also decreases. Okay, so next is fed state. That means if you have the drug after the meal, then what happens, that it impairs the progress of drug to in intestine. Okay, so due to presence of food in the stomach, the drug doesn't get into the intestine faster. Okay, and as a drug that doesn't get into the intestine faster, it decreases the absorption of such a drug. Okay, so this is again basically applicable to drugs which get absorbed in small intestine. If the drug is known to get absorbed in stomach, then it is actually a beneficial thing to have the drug along with the meal or just after the meal. Okay, so if there is increase in splanchnic blood flow, then there is increase in absorption of certain drugs like propranolol. Okay, so next factor is first pass metabolism. Okay, so you have already studied about first pass metabolism in biopharmaceutics, so I won't go into those details. So uh, drugs which get absorbed in gut wall like isoprenalin also have the effect of first pass metabolism. And also drugs uh, which get into the liver like opioids, beta blockers, and the nitrates, they also suffer from this first pass metabolism, and their bioavailability decreases. Okay, the next factor is presence of other agents like simultaneous or concurrent administration of other agents also have effect on the bioavailability of certain categories of drugs. So examples are given of vitamin C and the calcium. So vitamin C, if it is given with iron, then it increases the absorption of iron. And if iron is given along with the phytates, which are the some common constituents of some of the vegetables, then they retard the absorption of the iron. Retard means they decrease the iron absorption. Okay, so second example is the most common one, that is if tetracyclines are given along with the calcium, then complexation happens between calcium and the tetracycline, and thus it reduces the absorption of the tetracycline. The next factor is disease states. Okay, so if the disease related to any of the gastrointestinal tract is, if it is present, then it may lead to the malabsorption, then achlorhydria, cirrhosis, biliary obstruction. It can hamper the absorption. Okay, so malabsorption means what? If it is nothing but impaired absorption. Okay, achlorhydria, that is the hydrochloric acid, its production gets decreased. Okay, cirrhosis related to liver, again biliary obstruction, it is related to the bile duct. If there is something that can obstruct the flow of bile through the bile duct, then it is nothing but the biliary obstruction, and it is known to hamper the absorption of certain products. Okay, so lipophilic substances, they depend on the bile acids for its absorption to happen. Okay, so next factor is enterohepatic cycling. It means that the drug gets into the gastrointestinal tract. From there, it gets into the liver, and then it come back to the gastrointestinal tract, and this cycle continues. Okay, so this cycle is basically responsible for increase in bioavailability, and certain categories of drugs, they are known to undergo enterohepatic cycling. Examples are quoted of the morphine and the oral contraceptive pills. Okay, so let's see what is the concept of equivalence. So first one is the pharmaceutical equivalence. It means that equal amounts of identical active drug ingredient. Okay, that is the same salt or ester of the therapeutic moiety. So identical dosage form, if it is present, then it is not necessarily containing the same inactive ingredient. Okay, so pharmaceutical equivalence means what it requires is that dosage form should be same, and also the active pharmaceutical ingredient should be the same. Okay, so these two things, they if they are present, then, and bioavailability fraction if it is same, then we can say the two formulations are pharmaceutically equivalent. So one thing we have to focus on here is that inactive ingredients or the fillers, uh, or additives which we say usually, they, if they are different in both the formulation, it doesn't make any difference. What is important is that the active pharmaceutical ingredient should be same, and the dosage form should be same. That means if we take two brands of tablet containing same active pharmaceutical ingredient, let's assume omeprazole, then we are not concerned with what type of additives these two tablets have. We are only concerned with the active pharmaceutical ingredient, the tablet as a dosage form, and the equivalent bioavailability of the two brands. Okay, the next is pharmaceutical alternatives. What are those? So these are the identical therapeutic moiety or its precursor. So they, it is not necessary to have the same salt or ester of the therapeutic moiety, or the amount of it, or the dosage form it. Okay, so next is bioequivalence. So what is bioequivalence? That is pharmaceutical equivalent or alternative of the test product. When administered at the same molar dose, it should provide, or it should have the same bioavailability as the first drug. Okay, so it has the rate and extent of absorption as same as that of the first drug with which we are comparing. Okay, so there should not be any statistical significant difference from that of the reference product. So you should understand this, that in whenever bioequivalence studies are being carried out, So there is a reference product with which we are comparing our product. So a reference product will be the established product of the market, and we are comparing our product with that established product. Okay, so what is therapeutic equivalence? So same active substance or therapeutic moiety and clinically show the same efficacy and the safety profile. So you have to understand the difference between the uh, first concept and this concept, that is pharmaceutical equivalent. We are not talking about the safety and efficacy profile of it, we are considering the bioavailability of it. Sometimes this happens that two brands of the drugs, they may have the same bioavailability, but the efficacy and the safety profile may be different. It may happen due to the type of additives which are there in that particular brand. Okay, so this therapeutic equivalence means what? When we are comparing the two brands of the same active pharmaceutical ingredient, they produce the effect, clinical effect, to the same extent as that of the other product, and also they have similar type of adverse reactions or the side effects as that of the other product. Okay, so that is therapeutic equivalence. So we can see uh, whatever we have studied till now, it is like summary of it. Okay, so strength of dosage form, the excipients and other pharmaceutical factors, it relates to the amount of drug which is released from the dosage form. So the amount of drug which is released, it gets absorbed from the dosage form. So it gives rise to the amount of drug in the body. So concentration of drug in the central compartment, so it relates to the concentration of drug at the site of action. So it is like a distribution of the drug from the central compartment to the site of its action, and once a sufficient concentration of drug is present at the site of action, we get a response. Okay, so the amount of drug which get absorbed from the dosage form, it is affected by the patient related factors, and also it gets affected by the administration related factors. Okay, so again the same thing is there, but we have changed. It is in case of whatever bioequivalence or bioavailability studies with which we are doing. Okay, so in vitro quality control testing, it shows that amount of drug which is absorbed from the dosage form. Okay, then comes the in vivo bioequivalence studies. See the difference between two. In first, we are carrying out the in vitro quality control testing, by which we get to know that how much amount of drug it is getting absorbed from the dosage form. Then we are carrying out the in vivo bioequivalence studies, by means of which we get to know that is the amount of drug which is in the body. And then with the help of the PD studies, that is pharmacodynamic studies or the clinical trial studies, we are able to find out that how much concentration of drug is at the site of action, thus producing the clinical effect, and that we get from the pharmacodynamic studies and the clinical trials. Then what is the reference product? We have said again and again about a reference product. So let's see what is the reference product. So it is identified by the regulatory authorities as the designated reference product. So you cannot take any established product as the reference product. It needs to be uh, it needs to be mentioned into some records or some books of the regulatory agencies, that you have to use this product as the reference product. Then and then only you can take that particular brand of a product as the reference product. Okay, so it is usually the global innovators product. Okay, so it is protected by a patent. Then it is marketed under manufacturer's brand name. The clinical efficacy and safety profile is well documented in extensive trials. all generics must be bioequivalent to it. And in India, CDSCO may approve another product as a reference product. Okay, so whatever points we have studied till the last point, they are applicable to the global market. But in India, the CDSCO, which is a regulatory authority, can designate other local product as a reference product instead of a global marketer's product. Okay, so it is very much essential in case of the bioequivalence studies to get the reference product. Okay, depending on whatever reference products values are, bioavailability values are, we have to define the values of the generic product. Okay, so it should be comparable. There should not be significant difference between values which are associated with generics and with that of the reference product. Okay, so what is generic drug? So the drug product which is identical or bioequivalent to brand or a reference drug in these are the criteria in which it should be identical or equivalent. Okay, so first criteria is the same active ingredient in both, then route of administration is same, then dosage form is same, the strength, when we saying strength, we are actually talking about amount of active pharmaceutical ingredient in it. Okay, so that is the strength. So in case of paracetamol tablet, 500 milligram is the strength of that tablet. Okay, then indication means what, for what purpose that particular medication is being used. That is, in case of losartan is used as the antihypertensive agent. So if we are developing a losartan tablet, then the indication should be the same, that is for the antihypertensive purpose. And also safety profile should be similar to that of the reference product. Okay, so what will be the different thing in generic drug apart from the reference drug, that it may have different inactive ingredient, that is additives or the fillers, or it may have the different color of the product, and also it may have different shape. Okay, it is not mandatory to have a same shape as that of the reference product. It can come into different colors or the shape. Okay, so the color and shape, they just provide the aesthetics to the product. They actually don't have any role in the safety and efficacy of the product. So same thing is applied to the inactive ingredient. Why they are called as inactive? Because they don't usually they don't take part in the safety and efficacy profile of the drug. Okay, so almost half of the drugs in market have generics. Okay, so India being a house to the generic product, and it is a supplier of generics to the whole world. Okay, so we have huge generic market, and almost all companies of India, they produce the generic drugs. So that's why these bioequivalence and bioavailability studies, they matter the most in, or they are applied most for the Indian market. Okay, so the price difference between a reference and generic drug, so many of times it is being uh, it is being promoted by the branded drug products, that generic products, as they are cheap, the quality of ingredients that are used in those products, it is also not up to the mark. Okay, usually that is the uh, proposition, or the proponents of these branded drugs, they say about the generic product, but it is not so. Okay, when a company having a good manufacturing practice guideline it is following, then the whatever product that comes out of that particular company, it is of certain value. And you should know this very clearly, the company that manufactures the branded product, or it gets manufactured it from the some other manufacturing unit, same manufacturing unit produces the same product but by the generics name. Okay, so you should understand there is no difference in the quality. Only thing is that branded product, they are being promoted extensively, marketed extensively through network of medical representatives, but that doesn't happen in case of generic drugs. So whatever marketing cost is there, that particular cost benefit is passed to the consumers. That's why the generic products, they are cost effective as compared to the branded drugs. Okay, so let's see the difference between the generic and the reference drug. Reference drug, it is expensive, and the generic drug, it is 30 to 80% cheaper than the reference drug or the branded drug. So 5 to 5,000 new drug candidate tested in humans and one is approved. Okay, that's why it is expensive. But in case of generic drug, uh, they are just the copy of the reference drug. Okay, so they don't have to spend lot of money on getting the drugs tested in human beings or in animals. They just carry out the bioequivalence studies, and as they carry out the bioequivalence studies which are cheaper to perform, the cost is less. Okay, so the amount of time which is taken by the reference drug to get into the market, it is very much larger. That is, it takes around 12 to 15 years to get a drug molecule from the lab to the market. But in case of generic drug, the time is like a fractional time. In 6 months to 1 year max, the drug can get into the market. Okay, so the cost which is involved in getting the reference drug from lab to the market, it is very high. It is a cost around $1 billion, but the generic drug you can get into the market at a fractional cost of that of the reference drug. Okay, so next, in case of reference drug, the drug patents of 20 years applied before the clinical trials begin. Okay, so in case of generic drug, they are approved for sale after drug patent protection expires. So reference drug, it is protected for 20 years. In 20 years, they can make the money and get recover the expenditure which they have done for the research and development of the drug. And after expiration of these 20 years, the generic drug manufacturer, they rush to the FDA to get the priority license. Okay, so what is priority license? That is, they get 6 months of exclusivity period in which no other generic maker can get their generic product into the market. So the first one who gets the exclusivity period can get more, can reap the more benefits from the that particular generic product. Once after 6 months gets over, the other generic manufacturers can get into the market, and then the drug becomes commonplace. Then at that time, no one is going to pay premium for your product. Okay, if you are getting the same thing at a cheaper rate than the existing product, then at that particular time you will go for the cheaper rate product. You won't go for the higher cost product. Okay, so in case of reference drug, the if we consider that in a clinical trials, it takes around 6 to 8 years for concluding the clinical trials. So effectively, the reference drug gets 7 to 12 years of the patent period, where they are the sole owner, or they are the sole seller of that particular reference product. So they can keep the prices as they want. Okay, so a greater example of this is that sofosbuvir, a type of drug which is used for the hepatitis C, by the same company which have developed remdesivir, and that is under the brand name they have introduced the drug, that is Sovaldi. Okay, so the price they are quoting for this drug, that is $84,000. Okay, for 6 month treatment. So you can see that $84,000, it is very expensive. Okay, but still, as they are the patent holders, nobody can question them that why you have kept so much of price. Okay, so next is a fundamental bioequivalence assumption, that when a generic drug is claimed bioequivalent to a reference drug, it is assumed that they are therapeutically equivalent. Okay, so let's see some background to the bioequivalence studies. Using bioequivalence as the basis for the approving generic copies in US, Drug Price Competition and Patent Term Restoration Act of 1984, which is also known as Waxman Hatch Act, it actually paved way for this generic industry. Okay, so it created generic industry and increased their availability. So it is one of the most successful legislation in the pharmaceutical arena. Okay, so it benefited brands as well as the generic firms. So for generic firms, they rely on the findings of safety and efficacy of innovator drug after patent expiration. And also innovator firms, they can get patent extension of 5 years to make up for the time lost while their products were going through FDA's approval process. So after every clinical trial, FDA takes review of the things, so it takes time for these reviews. So innovator firms, they got a additional 5 years in order to make up the time which is lost during the FDA review process. Okay, so this particular Waxman Hatch Act, it actually helped the both, that is generic manufacturers and the brand manufacturers, and that's why this is the most successful legislation. So in case of Indian perspective, in India, CDSCO provides guidelines for bioavailability and bioequivalence studies, which is mentioned in Schedule Y. As per the Drugs and Cosmetics Rule, that is second amendment, which is done in 2005, all bioavailability and bioequivalence studies should be conducted in accordance to these guidelines. But some companies, they don't follow these guidelines and they tend to manipulate the data. So one company such, you may have known it, that is Ranbaxy. Okay, so it faces possibility of permanent injunction in US. Okay, so many of its facilities in that particular era, they were banned by the US FDA for flaunting the norms of it. That they, whatever data they have provided of the bioequivalence study, it was a falsified data, and that's why it faced many lawsuits in USA for the falsifying of data. Okay, so what are the requirements of BA studies for INDs or new drug application? That is the investigational new drug or new drug application. That is, in case of this, to establish equivalence between early and late clinical trial formulations. Okay. Second is formulation used in clinical trial and stability studies. Then clinical trial formulations and to be marketed drug products, and any other comparisons if appropriate. So second condition in which we require BA studies, it is ANDA. ANDA is abbreviated new drug application. It is for generic drug product. So change in components, composition and or manufacturing process also dictates use of BA studies. So if there is change in dosage form, like from capsule to tablet, then also we need to perform BA studies. So what is the objective of BA studies? Is the development of suitable dosage form for a new drug entity. Also, uh, it helps in determination of influence of excipients, patient-related factors, and possible interactions with other drugs. Okay, then Development of new drug formulations of existing drugs, we need to perform BAB studies. Okay, so also BAB studies, they help in control of quality of drug products, influence of processing factors, storage and stability. Okay, then comparison of availability of a drug substance from a different form or same dosage form produced by different manufacturers. So in these conditions also, the BABE studies, they will help, they will prove to be helpful. Okay, so when bioequivalence is not necessary, that is biowaivers. So in first condition, that is parenteral solution, that is same active substance with same concentration, same excipients, we don't require to perform bioequivalence studies. Then in second condition, that is oral solution which contain the same active substance with same concentration and also the excipients not affecting the GI transit or absorption, we don't require bioequivalence studies. So in case of gases, we don't require bioequivalence studies. Gases, as in case of the general anesthetics, we don't require to perform the bioequivalence studies. Then power for powder, sorry, not power, powder for reconstitution, such as in solutions which meet the criteria A or B, no bioequivalence studies required. So otic, ophthalmic, topical solutions, same active substance with same concentration, same excipient, no bioequivalence studies required. Inhalational products or nasal sprays which are administered with or without same device as a reference product, prepared as aqueous solution, same active substance with same concentration, same excipients, so biowaivers can be given. So a comparison between NDA and ANDA review process is done over here. What are the NDA requirements? So you can see till 5, that is till testing, it is same in NDA and ANDA requirements. But from 5 onwards, that is 6, 7, the 6th and 7th and 8th point, it is being replaced by bioequivalence studies in ANDA requirement. The first 5 points the ANDA can get from the NDA applications. Okay, so no need to again perform the same thing again and again, that is defining the chemistry, manufacturing controls, labeling and testing. Labeling can be altered, the controls can be kept same, the manufacturing process can be tweaked to little extent. But what is important is that we don't need to perform the animal studies, we don't need to perform any clinical trials, we don't need to perform any bioavailability study. Instead of it, we have to perform only one study, that is bioequivalence study, and which takes very much less time and very much less cost to perform. Thus the generic product, they are cheaper in price as compared to the reference products. Okay, so with this we have finished the chapter, that is introduction to the bioavailability and bioequivalence studies. Okay, thank you.